Okay, we are go, I think. Here we are.
All right, thank you everyone for zooming in with us. So today it's my great pleasure to introduce Will Towns, who is currently a postdoc in the Department of Computer Science at Princeton University, working with Dr. Barbara Engelhardt. And he's using probabilistic machine learning in studies of genomics, neuroscience, and ecology. But I want to go back to on the ecology part. So the interesting is Will's background. His undergraduate degree was in biology. And what I failed to talk to him about this morning was his following graduation. He was a Fulbright scholar at the University of the Philippines, where he conducted forest ecology research while living with an indigenous community. But when he came back to the States, I guess he was working in Virginia as a software analyst and then a software test engineer before deciding to go back and get a master's degree in math and statistics that he did at Georgetown University. From his master's, he then got introduced to some work and some people up at Harvard, where that's where he got his PhD in biostatistics. So he got that in 2019. He was working with Rafa Irizarry. And so now the work he's going to present to us today, the title of his talk is this non-negative spatial factorization. So Will, thank you for coming in. He's an applicant to our position that's open right now and help in the data science track.
Thank you so much. It's a pleasure to talk to you all about my research, but even more a joy to hear about what you're working on. And I've very much enjoyed my earlier meetings, and I know I have a few more later this afternoon. So thank you so much for hosting me. So I think this has already been covered a little bit, but yeah, I did not start out in biostats or anything like that. I was doing tropical ecology in the Philippines. This is the indigenous community, the Aitas, that I was, they were my guides in the forest. And this is the plant that I spent many hours staring at, one of many of that species. Fun fact about myself is my first statistics class I actually took at a community college as a night class while I was working full time. So I've had a very non-traditional path. Now on to the actual talk. I'm gonna mostly be talking about my method for my postdoc project, the non-negative spatial factorization. But for those who maybe come from other areas of biostatistics, I'll give a broad overview of single cell and spatial gene expression techniques, and where does the data actually come from, and what does it actually look like. And then, time permitting, I'll talk a little in more depth about the inference strategy that we've been using for scalable inference of Gaussian processes, which is broadly applicable as well to environmental data and other spatial data sources, and some of my possible future projects and other collaborations that I've been involved with. So there's three broad types of measurement strategies for gene expression that we've worked with over the years. Historically, RNA-seq or sequencing of gene expression was the new technology that surpassed microarrays. And now I refer to that as bulk sequencing or bulk RNA-seq. The reason being that you have to take a bulk sample of a tissue and slush everything up into a big slurry like a smoothie, and then you get a sense of gene expression in a tissue broadly construed. But you don't know about the differences in gene expression from one individual cell to another. So the newer technology of single cell RNA-seq and other single cell omics methods, there's a huge innovation to be able to profile the gene expression of individual cells. So that's represented by this fruit salad here. And even more recently than that, we now have the ability to actually profile single cell gene expression while retaining the spatial context of the cells. And as you can see in the single cell RNA-seq picture, the cells are disassociated. They don't have any kind of organization because you have to separate them out to do the sequencing. So my primary purpose of this talk is to tell you about some analysis methods for spatial gene expression, or spatial transcriptomics. When we're measuring gene expression, we don't actually know the expression levels of every gene exactly. We don't know the absolute counts of molecules. We take a kind of multinomial sample from the actual biological tissue, and this has important implications for the appropriate statistical models for the error. You know, I'm not going to go into all the details of the many complicated biochemical reactions that are involved in single-cell RNA-seq, but the most important part is the dissociation and single-cell isolation stage. And then also important to remember is that individual cells have a very small amount of starting material compared to a bulk tissue sample. So we need many more additional rounds of amplification through something like PCR to get sufficient material for doing the sequencing reaction. And the sequencing part is actually very similar across all three of these techniques. Here's what the data actually looks like. Obviously, this is only a small subsection of a data matrix, but single-cell RNA-seq are high-dimensional discrete count data. We have many zeros in our matrix. We have hundreds to thousands, even millions of observations of individual cells. So those are our samples, our observations. And then we also have a large number of features, possibly 20,000 genes in humans or mice. Of course, many of the genes carry no information, especially when you're looking at data within a tissue. So we're often interested in feature selection to narrow the set of genes down to only look at those genes that are interesting in some way. And depending on whether you are a statistician or a computer scientist, the way of representing the data matrix may be transposed. So in the R or statistics world, with gene expression, it's common to have the features in the rows and the observations in the columns. However, with Python and the computer science world, it's typical to use the transpose of that. So another thing to note is that individual observations may have globally higher expression levels across all the genes, and some genes have globally high expression levels across all observations. So you can already see some structure just in this very raw presentation of the data. And of course, the majority of entries in our data matrix are actually just zero. Here's a diagram showing how the one possible way that spatial transcriptomics data is collected. We could take a thin slice of tissue and lay it down on a slide or a plate that has numerous probes, much like a microarray, that will bind to the messenger RNA molecules. And each of the probes has a barcode to indicate its spatial position. So then once we've had this binding happen, we can then break things off of the slide and send them into PCR and into the sequencing. And the end result is we have a tiling of probes across the two-dimensional slice. And now, more recently, we're starting to have three-dimensional imaging techniques. And each of these spatial locations is associated with a high-dimensional gene expression count vector. And this is a schematic of what that might resemble. So the high-dimensional count outcome is very similar to single-cell RNA-seq, but now we also have associated with each observation a spatial coordinate of dimension 2, or mostly 2, but possibly 3 in the future. So here's an example of a very commonly used public spatial transcriptomics data. It's an interior sagittal section from a mouse brain. And here I'm showing the 0 being, a very low count of 0 being white and a high count being a darker blue color. And you can already see, for example, in this first gene, which I believe is the TTR gene, that there are certain particular spatial locations where it's very dark, but it's also very light in many other spatial locations. So that's a spatially variable gene. And it turns out that it's an indicator of the choroid plexus, which is a very spatially narrow region. So having given a broad overview of the data itself, I'd like to get into our approach to analyzing this data. So we have several goals. Dimension reduction is very important because, like I said, the outcome is a high dimensional count vector. We believe that genes interact with each other and are correlated. And so it's reasonable to assume that many genes may have a similar spatial pattern across all the observations. So we want to represent our outcome vector as some kind of combination, linear combination, non-negative combination of a smaller number of interpretable functional components, which I sometimes refer to as canonical surfaces or principal surfaces. And I'll show you some visualizations of those momentarily. We also might be interested in interpolation, filling in gaps where there was perhaps an error in the biochemistry. Think the probes didn't bind as well, or the tissue was wrinkled a little bit, or there was a tear or a hole, and we want to be able to create a continuous surface of gene expression across those gaps. And finally, we were interested in interpretation. We want to identify spatially variable genes. And for individual genes or individual observations, we want to compare and contrast the importance of spatial as opposed to non-spatial sources of variation. So I'm building up to our model. I'm going to show how it relates to several other probabilistic models for dimension reduction. So these are the desired properties that we'd like, to handle some of the things to answer some of the questions that I just mentioned. So first approach, which is widely used currently, is principal component analysis. Here I'm showing a probabilistic version of PCA, which the maximum likelihood estimator does converge to the actual PCA solution as you let the variance of the noise go to 0. So the advantage of this algorithm or model is that it's extremely fast and everybody knows what it is. So other than that, it doesn't really check many of our boxes, other than the fact that it does handle gene-gene correlations and provide dimension reduction. In prior work, I proposed the use of generalized principal component analysis, or what I call GLM-PCA, to appropriately model the count nature of the data. But for spatial transcriptomics, this is still not adequate because it's not accounting for the spatial correlation. Factor analysis is another very similar model to probabilistic PCA. The only difference is we're allowing a different noise term for each feature or gene. Now we're getting to something a little more interesting: non-negative matrix factorization, or what I call probabilistic non-negative matrix factorization, which essentially is very similar type of thing. It's just that it has a prior to do some regularization. Here we're actually achieving a non-negative decomposition of our high-dimensional data. And as I'll show with a simulation momentarily, the non-negative representation is highly desirable because it's more easily interpretable, and it tends to produce a kind of parts-based representation as opposed to a global representation. So all of the approaches I've described up until now totally ignore spatial coordinates. They're just generic dimension reduction, models for high-dimensional data. So now looking more from the spatial angle, we could think about looking at each gene individually and fitting some type of spatial Gaussian process across the spatial locations. And that's exactly what was done with the spatial DE method, which came out in 2018. And that was the start of Gaussian processes applied to spatial transcriptomics. More recently, there has been an interest in multivariate Gaussian processes, so that you don't have to look at each gene in isolation, but you can leverage some of these gene-gene correlations. And one approach that we propose, we're not the first ones to come up with this idea broadly construed, but essentially to have a small number of latent Gaussian processes with a mean function and a particular kernel function. And then we model the outcome as a Gaussian, where the mean is a linear combination of these Gaussian processes. So in the spatial statistics literature, this is known as linear model of co-regionalization. In neuroscience, it's known as Gaussian process factor analysis. So this is a very straightforward generalization of probabilistic principal component analysis or factor analysis to a spatial context. And there was another group that also came up with this idea independently, and they call it Mephisto. So I think I've already covered this. So I've not really described the Gaussian process itself. One of the most important things about a Gaussian process is the choice of kernel function. And a Gaussian process is a probability distribution over a functional space. So it's a distribution over the space of all possible functions over some spatial domain or temporal domain. Here I'm showing some examples in a one-dimensional, possibly temporal domain. And the form of the kernel function determines actually the smoothness of the family of possible functions. So a popular choice is the squared exponential or radial basis function kernel. It produces infinitely smooth functions. On the other hand, the Matern class, which is popular in environmental statistics and forestry, has a smoothness parameter. So if you let this, so this is actually 5/2, 3/2, and 1/2. And if you let the smoothness parameter go to infinity, it actually converges to the squared exponential kernel. So the smaller the smoothness parameter, the less smooth the class of functions. And you can see that immediately from this visualization of some random draws from this prior, from these different priors. The Matern 1/2 is actually also known as the exponential kernel, or I believe it's known as the Ornstein-Uhlenbeck process in applied math. But this is not very differentiable. For our application, we chose the Matern 3/2. We want to have some roughness because biologically, we believe that there could be sharp transitions in the gene expression profile between adjacent tissue layers. So we feel that the excessively smooth, infinitely differentiable squared exponential functions are too smooth, and they won't handle these sharp boundaries very well. And I'll show some evidence that that is empirically the case. But of course, choosing the right kernel is a whole area of research in Gaussian processes. So here's our actual model, non-negative spatial factorization. We're taking ideas from probabilistic non-negative matrix factorization and building in the spatial awareness through this Gaussian process priors. So we combine the Gaussian processes not through a linear combination, but rather through a non-negative combination. We also exponentiate them prior to this combination to ensure that they are also non-negative or actually positive. And just this small change dramatically alters the ability to do inference because this is no longer representable as a multi-output Gaussian process, and we don't have conjugacy, and we don't have a closed form solution to infer any of these unknown quantities like our loadings or weights, the W's, or the actual kernel hyperparameters or any of those sorts of things. So we have to develop a whole machinery to actually infer and fit these models. I'll just pause for a moment. Are there any questions about the definition of the model or how it relates to other probabilistic models? Okay. Feel free to ask questions anytime. I don't mind being interrupted. So here's just a summary of what I've just said. Here's just all of the different varieties of dimension reduction and spatially aware models. You can see that our NSF is really checking all the boxes. So we started off with this idea we want to be spatially aware, but then as we were working with some of our real data sets, which I'll show brief in a moment, we started to think, well, is it really fair to assume that all patterns of gene expression are spatially motivated? In fact, there could be cell types that are dispersed across a field of view that don't have spatial correlation. So maybe we need to also have non-spatial factors to model, I don't know, maybe immune cells or some glial cells in the brain that aren't arranged in a tissue architecture, but they're rather supporting actors that don't have a spatial pattern. And so that's how he came up with the idea of the non-negative spatial factorization hybrid model. We have essentially the same factor model as NSF, but we also bring in non-spatial factors here that are also non-negative. And because of the non-negativity, we can actually compute scores that give us a score between 0 and 1 for every gene or for every observation, telling us how spatial is this really, because we have non-spatial versus spatial. So here's a simulation where we know the ground truth. We draw some genes from 4 canonical patterns that have a spatial arrangement. And then, we use a negative binomial draw to just create some noise and make it a little bit of a challenge to infer. And if we apply a method such as Mephisto, which uses real valued latent factors, or our implementation using real valued spatial factorization, both of these have real valued factors. The goodness of fit is excellent. Prediction is fine, but the interpretability of these latent factors is very limited by the fact that every factor is sort of a jumble of all the components. So, if you look at the simulated data, you can see the truth is we want one component with 4 and another one with this diamond shape and so forth. But here they're sort of convolved together. And in the original non-negative matrix factorization paper, they also observed this phenomenon comparing NMF to PCA. And they looked at faces as the data they want to decompose. And PCA would learn eigenfaces. So each principal component represented a kind of average of many faces, and you could take linear combinations to make predictions for individuals. On non-negative approach, each component was a particular region of the face, like an eye or a nose or an ear. And so that's what we're really interested in here. And as you can see, our approach, non-negative spatial factorization, was able to successfully recover these and sort of deconvolve these various spatial patterns. So hopefully that motivates the importance of the non-negativity constraint. Okay, so now let's look at some real data. I have 3 example data sets here with different characteristics. We had a SlideSeq version 2 with over 30,000 spatial locations. Here the scalability was critical, and it has essentially single cell resolution in terms of the spatial resolution. We also had XYZeq, which had a much coarser spatial resolution and multiple cells at each location, as well as Visium, which was essentially intermediate between those 2 extremes. And I already showed you the histology of the Visium slice. For the prediction challenge, we wanted to see if we fit these models and try to evaluate its accuracy on held out data. So we randomly removed some of the spatial locations, and then we fit the model on the training data and tried to make predictions on the held out data. What we found was that using real valued factorization is actually really good for prediction. And in fact, the best performing one was our RSF, real valued spatial factorization. And it was interesting that it actually has a very similar probabilistic structure to Mephisto, but greatly outperforming in terms of the accuracy. And our explanation for this is we believe this is coming from the choice of kernel. The Mephisto model uses the squared exponential kernel, whereas we use the matern kernel. And we think this ability to handle the roughness and the discontinuities in the data is better handled by the matern kernel. Among the class of non-negative models, we found that including the spatial information did improve the accuracy. So PNMF is not spatially aware, but these are our 2 models that did leverage the spatial coordinates. Here's another data set where we found totally different results, where we actually found that our non-negative spatial factorization did just as well as the real valued spatial factorization. But again, we see a very dramatic improvement in accuracy by choosing an appropriate kernel, matern kernel function. And we also see that we're improving on the spatially unaware factor analysis and NMF type of approaches. In terms of speed, of course, nothing can beat PCA or its probabilistic cousin factor analysis. using Gaussian processes is inherently more complicated and it's going to be computationally slower. Our main goal here was to see that our computational performance was comparable to the other, state-of-the-art method, which was Mephisto. And we were pleased to see that we actually had a better ability to handle larger data because Mephisto frequently ran out of memory. It wasn't so much the time as the memory constraint. So we're able to handle these memory issues a little more efficiently. Another interesting thing we wanted to check on is the sparsity pattern of our weights. So these are some of the loadings matrix to define the relationship between features and components. And what we were pleased to see is that increasing the number of latent components or number of dimensions of the model actually led to a higher degree of sparsity. So because we're using these probabilistic models, this is showing the regularization effect of using these Bayesian priors. And of course, real valued factor models had essentially no sparsity. Even though Mephisto is a real valued factor model, they explicitly promote sparsity using a spike and slab prior. But we were able to, to achieve a reasonable degree of sparsity as well, simply by having a non-negativity constraint. We didn't do any additional priors to encourage sparsity. Of course, that could be a future extension of the model for those who really care a lot about sparsity. So here's a visualization of some of the what I was referring to as canonical surfaces or principal surfaces learned on the 30,000 plus observation SlideSeq2 mouse hippocampus. This is for those who are interested, this is a coronal section. And what we were excited to find is that these non-negative spatial factors could really zero in on extremely specific regions of the brain. And so, I'm not a neuroanatomist, but my understanding is, so this one, for example, is the thalamus. And this here, I think, is the choroid plexus. This one is the medial habenula, the dentate gyrus. My favorite one is number 10, which you can see is extremely thin and narrow layer of tissue. So this is actually the meninges, which is a very small, narrow wrapper around the outside of a brain layer. And the way that we can identify these, other than consulting a neuroscientist, is we can actually look at which genes are loading most strongly onto each of the latent components. And then, those genes are often markers for particular cell types or for particular tissue regions in the brain. So here I'll show a visualization of that. So for each component, we found the top most loaded gene, and we just plot the expression pattern of that one gene across space. And what you can see is that there's a strong correspondence between these marker genes and their spatial regions. However, the individual gene's expression pattern is much noisier than the latent factor representation. And the reason for that is genes are inherently stochastic. And if you just look at one gene at a time, you're going to get a lot of noise. Just from biology, you're going to get noise, not even to mention the technical noise. So hopefully this really motivates the importance of a multivariate approach where you can leverage each component being learned by many genes that load onto it rather than just a single marker gene. When we look at non-spatial factors from the hybrid model, we can generally see that they don't have very obvious spatial patterns, as you would expect, because there's no spatial information being used by these factors. So that was just kind of a sanity check. Here's another data set. This one's from a tumor model in a mouse liver. And there's many fewer spatial locations, so it's a little bit harder to see. But for example, this spatial factor number 1 ended up tagging hepatocytes in a normal tissue region of the liver, whereas the factor number 3 was really tagging a tumor region. And our final data set with the 10x Visium, again, you can see characteristic brain regions being pulled out in an unsupervised way by these spatial factors. So for example, here we have the cerebral cortex, the basal ganglia, These are fiber tracts like the corpus callosum, the inside of the olfactory bulb, and the choroid plexus. Of course, it's not guaranteed that every spatial factor will exactly match to a single tissue layer. This is an exploratory tool. Of course, you're going to have to do additional validations, but it's meant to suggest ideas and suggest hypotheses that could be followed up with more fine-tuned approaches. I think I'll skip over this in the interest of time. So now I want to think about how to quantitatively assess the importance of spatial information as opposed to non-spatial information. So if you are a cell, is it more important what your cell type is, or is it more important what the expression patterns of your nearest neighbors are? So in the non-negative spatial factorization hybrid model, we have a spatial component, which has its set of loadings, and we have a non-spatial component, which has its set of loadings. So if we basically look at W over W plus B, I mean, it's more complicated than that, but that's the general idea. Because these things are non-negative, that ratio is going to be a number between 0 and 1. And if it's 0, then that means that essentially there's no spatial information that's relevant to that gene, that the gene is wholly explained by the non-spatial components. And if it's 1, then it's the other way around. And you could do the same thing with the observations. So once we've computed a spatial importance score for each feature or for each observation, we can make a histogram because there's thousands of features and thousands of observations. And what's interesting is we found that there were particular genes that were completely determined by spatial information. They're strongly spatially variable. On the other hand, there are some genes that basically spatial information doesn't tell you anything about that gene. It's just active in a dispersed way across the field of view. And for observations, there's certain observations that are very strongly determined by the spatial context, and other observations, maybe not so much. So once you have these scores, you can of course follow up and say, well, what actually are these genes that are so strongly spatially variable? And let's look into the data. And we did this a little bit in our paper. Let's go and look at Go terms, and let's look at other databases to assess what's already known about those particular genes. So now I'd like to talk a little bit more. I'm probably going to skip over some of this in the interest of time, but just to give a flavor of the strategy that we use to achieve scalable inference for these Gaussian processes with such large numbers of observations. So for those who are familiar with Gaussian processes, the compute time is O of n cubed, where n is the number of spatial observations. And this qubit complexity is very undesirable when you have tens of thousands or hundreds of thousands of observations. So it's a long-standing problem in the spatial statistics literature, and there's a plethora of different approaches. The approach that we're most excited about from the statistics literature is the nearest neighbor Gaussian process, which was developed by Sudipto Banerjee, who I think is actually at UCLA, and many of his students and collaborators. And then in the machine learning world, an alternative approach called variational inference using inducing points has been developed. So our implementation currently uses the variational inference strategy. We are actually currently working on unifying it with the nearest neighbor strategy because they both have strengths. Bayesian inference, this is just a review, hopefully for most of you. But we have a likelihood, such as a Poisson distribution for counts. We have a prior, which maybe is some factor model involving Gaussian processes. And what we really are interested in is the posterior distribution of our latent factors and over conditional on the observed data. But we're not able to compute this high dimensional integral in closed form. And even if we could do it in closed form, it would not be computationally tractable. So the strategy that we follow is we basically are going to sample from some tractable family of distributions, which are called Q, and we call this the approximate posterior. And we're going to parameterize this family of distributions And then we will perform optimization to move around within this space of distributions, or parameter space, since it is a parametric family, until we find an optimum subject to the constraint of tractability. And this is all very abstract, but this is the general notion of variational inference. And the way that we quantify how good a distribution is we essentially measure the KL divergence from the true posterior to this approximating distribution. So here's an example of this where we're saying we want to fit a multivariate Gaussian, but we're going to constrain it to be the space of isotropic Gaussians, and we will move this, these spheres around until they center, in the actual mean, and we'll adjust the variance to somehow be the best possible approximation. So we're not getting the true posterior, but we're hopefully getting something that's relatively close, subject to tractability. So I don't think I'm going to go into too much detail about this. This is actually the way that we formulate the objective function that we want to optimize. So unlike other forms of Bayesian inference where you need to do Markov chain Monte Carlo sampling, which is very time consuming and slow, we can actually represent this as a straightforward optimization problem. And we can use all of the techniques from deep learning, such as stochastic gradient descent and mini batches and all momentum optimizers and things like that. So just briefly, to give a sense of how the inducing point strategy works, it's more or less analogous to a regression spline strategy where we have knots or inducing points scattered throughout the field of view. And then we're going to condition on those and then infer the distribution subject to those knots. So our complexity is reduced from O of n cubed to O of m cubed, where m is the number of knots. And here's an illustration of what this looks like. We start off, this is not my simulation. We start off with some kind of noisy approximation, and then, the white circles are the knots. So you can see that even with a small number of knots, we can actually achieve a pretty good degree of accuracy. And this is my data. This is the Visium mouse brain slice. And in the way that we initialize the knot locations is simply to apply k-means clustering to the spatial coordinates. We ignore the gene expression. And this essentially spaces them out equally it's more appropriate than using something like a regular grid, because the boundaries of the tissue are not regular. And here you can see the computational effect of different numbers of inducing points. So we tried various numbers of inducing points, and of course the complexity and the time increases with larger numbers. But with larger numbers of inducing points, you also get a much more accurate approximation to the true posterior. So in practice, we had to solve all kinds of engineering challenges, Cholesky instabilities, how to even detect convergence of a very noisy objective function. And I'd be happy to talk more about that in detail with anyone who's interested. If you want to try our method, we're still working on the Python package. You can install it, but we need to write tests and we need to improve the documentation. But it's publicly available for those who want to experiment with the code. It is implemented in TensorFlow and Python. I'll skip over this to save time. I think I'll also skip over this to save time. So I'll briefly talk about a few future projects that I'm interested in, in addition to, continuing on with some of the themes that I've covered today. So we're still a little bit limited with the scalability. We've gotten up to the tens of thousands of observations. This inducing point strategy is not going to scale to millions of observations. So that's why we're interested in this nearest neighbor approach. But the implementation is much more tricky. And so that's really what we're focused on right now is, can we combine the variational strategy, which is needed to deal with non-conjugate likelihoods like the Poisson, with the scalability to truly huge data using nearest neighbor Gaussian processes. So here we have an example from the literature where a nearest neighbor Gaussian process is applied to very large scale forestry data. So we believe that a lot of these ideas can also be brought into the genomics world. Another really cool area is how can we actually define a Gaussian process on a manifold as opposed to just a straightforward, kind of Euclidean domain. There's a lot of really exciting work being done in this area. I really think that manifold-aware Gaussian processes will be necessary for certain tissue types. Here's an example from embryo, where you can see that something that's close in Euclidean space is not really close in biological space because there's this gap between the tissue layers. So that's something that would be good future work. I also have kind of a side interest in wearable devices, and I think some of the ideas from spatial statistics could also be applicable to temporal signals and high dimensional sensor data. And I guess we have about 2 more minutes, and then I'll let there be questions. But just to give a flavor of some of the other projects that I worked on over the years, I've been heavily involved in methods development for single-cell RNA-seq, feature selection, dimension reduction, quantile normalization. Very passionate about open source software and be in the form of R packages and Python packages. So you can check out some of those that are all publicly available. I have a lot of side interests. I'm interested in infectious disease. I've done some collaborations on Ebola modeling and neuroscience, as well as longevity. And I think I will skip over these. If for folks who are interested, I've been mentoring a couple of PhD students, and I'll be happy to also talk about the work that they've been doing. But I think we're getting a little short on time. So thank you to my collaborators and advisors, and thanks to all of you for your time and attention. I'll be happy to take any questions.
Thanks, Will. That was great. Does anyone have a question for our speaker? So just unmute yourself.
Hello, Will. Hello, this is Eleanor. I'm a second-year PhD student in Biostats. And I just want to say this is very impressive work. And I really like the way that you put together those, like a brief review in terms of how those different kind of models are compared to each other in the beginning. And so I have 2 questions. So the first one is, so is this model have a identifiable issue in terms of those loadings and the factor in the latent space?
Right, that's a great question. So all of these factor models are unidentifiable when you're talking about point estimates. And so if you wanted to specify one of these models and then just try to learn like the maximum likelihood estimator and dispense with all the Bayesian machinery, then yeah, things are going to be unidentifiable, much like a finite mixture model would be unidentifiable up to label switching of the clusters, right?
Yeah.
So with PCA, for example, the maximum likelihood estimator is going to be more or less unidentifiable up to a rotation of those factors. And you could say the same thing about a point estimate for NMF. It's invariant up to, I believe it's called a monomial transformation. It's essentially a permutation of the ordering of the columns and rows combined with a rescaling. Like if you divide factor 1 by alpha and then you multiply loading 1 by alpha, then it cancels out. So all of that goes away when you have a Bayesian prior, because it enforces this to come from a probability distribution. So I think it's not something that we really worry about. But yeah, we don't have any theoretical guarantees about these estimators. Our objective function is a stochastic function, meaning that we can't actually evaluate the objective function without a sampling procedure. And the solution is likely to be a local optima, because everything's non-convex. So those problems are actually an even bigger problem than identifiability, which it's not that it's not a problem, but there's just so many even bigger problems that we haven't even gotten to the point where we can really get into those kinds of theoretical questions about identifiability.
I see. And so another question I have is, so how the model determine the number of latent factors in there? Like, or are you gonna just overestimate it in the first place?
Right. Yeah, that's a huge question with any of these types of models. For a while, I was talking to some of the professors earlier about Bayesian nonparametrics, and if you really didn't want to have to choose a number of components, you could think about some kind of like Indian buffet process or something to actually learn the number of latent dimensions. Unfortunately, those Bayesian nonparametric approaches are even more intractable, computationally and inferentially. So yeah, so we have to face a trade-off here. Yeah, you just have to try different numbers of components, and we would fit these models with 6, 12, 20, 30, and just kind of look at it and see if it gave us any information. It's a very ad hoc, exploratory type of mentality. But to be fair, you would have the same exact problem with principal component analysis.
Yeah, exactly.
You can make plots like scree plots or proportion of variance explained or things like that. But unfortunately, it's difficult to be rigorous about that question.
Yeah, definitely. And just lots of comments. So I'm totally with you in terms of the scalability of VI, because I've been using this factorized model, and we're using VI as well.
Cool.
So yeah, it takes a long time to run for now. Yeah, I know. And thank you for the talk. Yeah.
Thanks for the great questions.
Anyone else?
I have a question. Very nice talk.
Question, please.
So in other applications, when count data are analyzed, often overdispersion was the issue that people have to deal with. I wonder if for single cell data, is overdispersion ever an issue?
That's a great question. And this is something that has been much debated. In fact, I just saw a paper came out from, I think it was from Rahul Satija in one of his postdocs. So our GLM PCA paper was all about convincing people to not use Gaussian models for count data, and to not think that they can do transformations, to transform their way out of trouble, so to speak. And it's especially a problem when the counts are very small. If you have large counts, then you more or less could say, well, okay, a Poisson, looks like more or less a Gaussian when you have a large mean. But with single cell data, often the mean is very low. And so the Gaussian approximation to even a variance stabilized Poisson or negative binomial counts is very poor. So that was really our goal. But then after I think we were fairly successful in convincing people that you need to think about the count nature of the data. Then it moved on to, well, should it be Poisson or should it be negative binomial? And there's a raging debate still going on about which of those is more appropriate. And if you use negative binomial when you're fitting a factor model or something like that, should you have one dispersion parameter that's shared across all genes, or should you allow each gene to have its own dispersion parameter, or should you use some kind of hierarchical model to shrink those towards a common value? Personally, I do think that there is over dispersion. I think that the technical noise from the sampling and the bead capture and all of that is adequately characterized by the Poisson, or really I think it's a multinomial process, but I think the Poisson is a reasonable approximation to the multinomial. But if you're also going to take into account the intrinsic stochasticity of biology, the bursting nature of transcription, then yes, there absolutely is overdispersion. And if you want to absorb that overdispersion into your error model, then you need something like a negative binomial or maybe a Poisson log normal to account for that. And I think there has been more evidence supporting this need for overdispersion recently. Unfortunately, the downside of that is fitting a negative binomial regression model is it's not going to give you that very convenient closed form solution like a Poisson GLM might have for doing things like feature selection. And so I think there again is the trade-off between accuracy and appropriateness of a model versus something that may be quick and good enough. So definitely think that depends on the type of application. But yeah, I think that yes, there is overdispersion. That was a long answer for a simple, should have been a simple answer, but I love talking about that. It's a really interesting thing to debate.
If I can follow up, I'm sorry, am I unmuted? Yeah. In the microbiome literature, there's commonly used as the zero inflated negative binomial. Given all of your zero counts, is that something that you would need to consider?
Oh, wow. I could talk about that for another hour. That actually was part of my earlier work with GLM PCA. So my belief, or not belief, the evidence supports, in my opinion, that that for molecule counts where you have unique molecular identifiers, which are basically barcodes to remove duplicates that result from PCR replications of molecules. If you have the molecule counts, which most current single cell protocols do have those, that actually there is no zero inflation. You see a large number of zeros, but the mean is also very low. So there's not an excess of zeros relative to what the mean is. So in read count data where you don't know the molecules, then yes, you do absolutely see zero inflation, because you have, for each molecule, you have many PCR duplicates and many reads that are just coming from a single molecule. And so that shifts the mean to be much larger, but you don't duplicate and replicate the zeros. So you end up with a big spike at zero and something that looks sort of log normal for everything else. So if you have to analyze those data, then yeah, you might need a zero inflated model. What we actually argued in another one of my papers was that it's actually more straightforward to apply quantile normalization to try to computationally remove some of those PCR duplicates and shrink the distribution back to something like a Poisson or negative binomial, and then to apply a non-zero inflated factor model, for example. So I think in microbiome, which I'm less familiar with, it would all boil down to the question of, are you looking at molecule counts or are you looking at read counts?
I have another question. So you mentioned spiking nature of single cell gene expression. Now you have spatial data. I'm just curious, have you observed whether spiking tends to be random spatially or clustered?
Yeah, so that is a really great question. I don't think the type of data that I myself have been looking at can answer that question. I think what you really want there is spatiotemporal data, like imaging data. And there has been some work by Jared Tocher's group at Princeton, and one of my colleagues or lab mates did a collaboration with that group where rather than looking at a genome-wide profile of expression for every gene, you instead focus on one or a handful of target genes where you can actually look at in situ measurements of expression over time. So you have these movies of cells. And I believe the gene they looked at was called ERK, if that rings a bell. This is not really my main area of expertise, but I think it's very interesting. And so what they did was they developed a point process to model whether, to what extent, if you're a cell and your neighbors are all spiking this particular signaling gene, whether that leads to an increased probability of the firing rate of that cell itself, or whether the firing rate is intrinsic to the cell and not really sort of agnostic to the firing pattern of its neighbors. And I think they were epithelial cells. They're not neurons. So the similar types of questions are being asked with like neural, like spike train data. But I don't have the answer, and I don't think the exact data that I've been working with could answer that. I think you want the temporal component there to think about the causality and things like that.
Thank you.
Yeah, thanks very much. Any last questions? It's almost 1 o'clock. Thank you, Will. It's been really wonderful, thought-provoking material.
Hey, Will. Really cool talk. So I had one question regarding, so you mentioned manifold learning or like some latent factor models that learn directly over manifolds with like some applications towards like either single cell data or some like biological data. And so, do you have like a sense of like, how much like in terms of like a specific manifold for the data or like something like a target, like how much that can be informed from like the particular process? Like if we're, for example, if we're thinking about like single cell data, right? Like, is there like a particular manifold that we expect, like some low dimensional manifold that we expect the data to be sitting on that is structured in a way that would be different from like if we, wanted to investigate like bulk RNA-seq data, right? Or if we had like spatial transcriptomics or something.
Sure. So I think what you're asking about is manifold learning for, if you're talking about single cell RNA-seq, that's actually a whole another area of research. Maybe you don't quite have time to get into. The specific example that I'm referring to is where the manifold in the sense that the spatial locations, you can see that there are tissue layers and there are gaps, and you don't want to assume that the correlation in gene expression is simply a function of Euclidean distance. You want to take into account the fact that, you need to be a nearest neighbor, but you can't be a nearest neighbor if you're across some gap in a different axis of spatial variation. So, so that's a very different problem where you actually do know, you can know the manifold before you start developing your model. Trying to learn a manifold is much harder. And of course, there's work with Gaussian process latent variable models and things like variational autoencoders that try to learn these nonlinear, surfaces, or even projecting onto hyperbolic spaces. And it's very interesting. But I don't, I haven't done research in that area myself. I just have a familiarity with it. So, maybe we could talk offline. I can point you to some really cool papers if you're interested, or, that sort of thing.
Cool, thanks.
All right, well, thank you very much. That brings us to 1 o'clock. So appreciate your giving seminar and visiting us today, Will. I think we have an afternoon set up. Hopefully you have breaks during your schedule too. But thanks, everyone. We will meet again next week. Thanks.